Hi students, welcome to Year 11 Biology and Module 4 Ecosystem Dynamics. This is video number 11 and we're going to look at gas analysis. In this video, we're going to look specifically at investigating and analysing past and present technologies that have been used to determine evidence for past changes, and specifically in the context of gas analysis. So we've kind of mentioned this previously when we've talked about things like ice cores and the fact that ice cores have been used to extract air in a vertical column that allows us to go back in time. If we have an idea of the rate of formation of the ice, then we get a little bit of an idea as we go back in time of how old that is, and also we can use other markers such as particles that are trapped in the ice that may have radioactive materials in them that allow us to produce some dates for particular sections or layers within the ice cores. But within the ice too is air bubbles that are trapped, and that can also give us something of a clue as to the concentration or proportions of different gases in the atmosphere at a particular point in time. To do that, we need to extract the air from the ice cores. In a vacuum, we need to be very careful with this procedure that it doesn't contaminate, or that the samples are not contaminated by our current atmosphere, because obviously that would seriously affect the results. So this can either be done by crushing or by melting. Often crushing is more successful, it's more of a dry extraction method, and it's very important for things like carbon dioxide. We want to make sure that the ice is cooled and the crushing doesn't create that sort of wet molten water that we get as a result of that heating of the ice. And this allows us to release the carbon dioxide bubbles and then use gas chromatography in order to determine what the components of the gas are. The gas chromatography method is very similar to probably more liquid chromatography that you'd be more familiar with. So, and that's usually people have done things like put color textures on a piece of filter paper or chromatography paper and allow the fluid to basically move through, and the colors will then separate out of the texture. Same sort of thing can happen with pigments in leaves. And this allows us to separate different components of a mixture based on differences in the physical and chemical properties. And one of those very important properties might be just the difference in mass between different components of the mixture. But it can also include things like absorbance. In doing analysis of things like ice cores, we get a chance to examine some differences between the different forms of oxygen that can also exist. Now, oxygen is obviously associated with the gas oxygen, the diatomic gas O2, but oxygen is also present in two very, very important compounds that are also going to give us a little bit of a clue about what's going on in past atmospheres. That is carbon dioxide, CO2, and water, H2O. What we know is that oxygen 18 can also exist when we're separating the components of the mixtures by their mass. So oxygen 18, 8 protons and 10 neutrons, compared with oxygen 16, 8 protons and 8 neutrons. The proportions of these two isotopes in ice cores can give us an idea about past global temperatures, especially where there has been warming or cooling events, because this affects things like the rate of precipitation, evaporation, and the fact that the oxygen 18 isotope is a little heavier means that it is more likely, or less likely depending on the temperature, to be evaporated from the ocean, so that can change the mix in the oceans, and also to fall as precipitation, rain or snow, and to form those very important ice cores. So tracking the ratio of oxygen 18 to oxygen 16 is one very important marker of ancient water temperatures, and also a clue to things like greenhouse events or icehouse events that might have happened in the past. One of the things that we'll do in class is we will have a look at some data, because this section is really about analysing data. So we're just providing a little bit of a snapshot underneath, just so we understand where this data is coming from. And I guess the most important thing that we need to talk about when we're talking about the gas analysis is the way that gases have been analysed over time to suggest that the early Earth's atmosphere was different from the current one. So this, a very famous experiment, the Urey-Miller experiment, was done to try and duplicate the atmosphere of the early Earth and to try and use that to see if there was the possibility of producing organic molecules. Now, when you look at the early Earth's atmosphere, one of the things that you notice is the absence of oxygen. We know that the Earth's current atmosphere contains over 20%, so over a fifth of its volume is oxygen. But in the early atmosphere, it was very, very low. So this was going to have consequences on things like the concentration, not just of oxygen in the atmosphere, but the concentration of ozone. And we know that ozone has a very important role in the layer above the layer that we breathe. So the troposphere is basically the layer of Earth that we breathe. The stratosphere is the level above that. And in that kind of intermediate area between these two at the bottom of the stratosphere is the ozone layer. And the ozone layer is particularly good at filtering out wavelengths of ultraviolet radiation. And so that without that there's a much higher proportion of ultraviolet radiation that's going to reach the Earth. And that's going to create a very inhospitable environment for organisms. But as we find, particularly through evidence from the banded iron formations or the red beds, we find increase in oxygen levels, which led to the precipitation of iron oxides. And these iron oxides were the beautiful red color, and they were deposited in some of the rocks, and we can see that today, possibly in the oceans where a lot of the oxygen was being produced by aquatic organisms. Similar to the stromatolites that you can find in Shark Bay, so the cyanobacteria that build those beautiful stromatolite structures. And we find fossil stromatolites that suggest that we have the same sort of organisms existing today doing a very similar job to what those ancient organisms were doing. And that is releasing oxygen through their body processes, through their cellular processes into the oceans, and therefore building up the levels of oxygen. Now, the levels of oxygen would have increased in the water before they were released into the air. So we do find that some of those precipitation effects were happening around things like the iron oxides precipitating out before we also had that increase in oxygen in the atmosphere that was allowing us to do things like form the ozone layer, protect a lot of those early organisms from that those high doses of ultraviolet radiation, and ultimately lead to some of the diversity of life that we see on Earth today. Analysing gases is all about looking at tables and graphs and trying to draw some conclusions about what the key gases were and how the quantities of those, or how the proportions of those, have changed over time. So certainly with things like nitrogen and oxygen, and also potentially some of those more volatile gases, ammonia or methane, which were part of the early atmosphere and seem to be in far less quantities or proportions in the current atmosphere, have created some interesting data for us to use to compare, to have a look at what ancient environment atmospheric conditions may have been like. Thanks for watching.